I'd like to tell you about one of the most exciting areas of research that you could possibly pursue in our country. This is about our biodiversity and the discovery of new drugs from marine organisms in the Philippines. As you know, Philippines is the center of marine biodiversity in the whole world. Look at this cartoon of an evolutionary tree. You must know that life originated from the seas. Now, in the Philippines, we have the richest marine biodiversity in the world. My research focuses on one of the earliest organisms that evolved in the sea. And this would be the sponges, which are the first multicellular organisms or metazoans. My other organism of interest are the mollusks. So take a look at where they are in the phylogenetic tree, and later we will be able to correlate this with the kinds of compounds that are produced by these organisms. So what's the basis for the research? It's evolution, it's ecology, it's biology, which provides us the chemical leads. We say that biodiversity is the source of chemodiversity. Sponges, snails, and shipworms, which are also snails, and their associated microorganisms or microbes produce small and medium-sized molecules to ward off competitors, to ward off predators, and also to catch their prey, or simply to prevent overgrowth on their surfaces. Now, this would be the ecological basis for discovering new drugs for infections, for pain, and for cancer. So, why do we need new drugs for these diseases and human conditions? That is because the drug pipeline is running dry for infectious diseases, for cancer, pain, and because we have an aging population, for aging and neurodegenerative diseases. Now, drug companies in the world have invested so much money to test what we call combinatorial chemistry libraries. These are synthetic libraries, but they have not been very successful about discovering new classes of drugs. This compared to the age-old, time-tested source of drugs, which is nature. This is what we call our biodiversity libraries. You must know that 60 to 70% of all drugs used in the clinics today are of natural origin or were derived from natural compounds. So this is my area of research. It's called Marine Natural Products. And here we have a group of young researchers working with me, young faculty and graduate students, as well as technicians. Currently, I have 4 major research programs and projects. The first is the International Cooperative Biodiversity Group Program, funded by the U.S. National Institutes of Health, and it's titled Philippine Mollusk Symbiote. We are studying the bacterial symbionts of mollusks. And the program leader of this ICBG program is Margot Haygood of the University of Utah, and I am an associate program leader.
The basis of the project is the discovery of new drugs from Philippine biodiversity. So what is biodiversity? Biodiversity refers to all the different types of organisms, plants, animals, and bacteria that live in an area. And the Philippines is exceptionally diverse, and so it has a wealth of opportunities for discovering new drugs. So our project studies mollusks. So what is a mollusk? Mollusks are familiar to humans in the form of food. So all of us have eaten clams and mussels and squid and other foods like that. Those are all mollusks. Drugs from the 20th century were found made by bacteria that live in the soil on land. But now in the 21st century, we're starting to look at the sea. And then what we do is we isolate bacteria from these mollusks and we study the mollusks in the laboratory.
So after they culture the bacteria for small scale, here in the chemistry lab, we do the small scale extraction. So we centrifuge and isolate all the secondary metabolite from the bacteria. And then after that, we submit the extract. So here are some of the samples of the extracts that we get from the chemistry. And then we dry them using our CentriVap and RotaVap here. And then after we dry them down, we archive all our extracts here. So here are some of the samples of our dried extracts in the vial. So we take the weights of the vial and then we submit all these extracts to various assays like our anti-infective assay, anti-cancer assay, and neuroassay.
Then we have the DOST funded project to discover anti-pain and anti-neurodegenerative agents. With it, I'm studying conus and turids, the venoms, also for neuroactive peptides, with Dr. Baldomero Oliveira of the University of Utah.
So here we are already inside the snail room wherein as you can see the different species of conus snails, or this is actually the main focus of the discovery and development of health products, DDHP project, wherein we dissect those conus snails and using the venom duct we extract those venoms in order to have the bioactive fractions to be tested on different set of assays.
I'm not only studying marine biodiversity, I'm also studying terrestrial biodiversity. I have this special interest in cancer, which I have published on many years since I started my research. This time I'm working on moringa or malunggay. And I'm looking at anti-tumor agents and cancer preventive compounds from moringa.
The bioassay guided fractionation method wherein we fractionate the different extracts from the moringa plant and then test it on different bioassays. So the moringa project have different bioassays. So the first part is to test the in vitro cytotoxicity of the moringa extracts. Then the second part will dwell on the possible mechanisms of action of the extracts.
The last item in this list is synergistic combination of the drugs that we have isolated from these plants and marine organisms for cancer. So think of how you can combine a drug in the clinics with a new compound that you've discovered from plants and marine organisms, and maybe there's a way for the two compounds to act synergistically to extend the life of cancer patient. So let's not forget that first we must ask permission from the local communities who are the stewards of this biodiversity. So my group has pioneered in getting the permits to undertake sample collection from marine sites. So in the last MOAs we got signed with the DAB-FAR were for the ICBG program and for the DOST project. The principle being that First, we must get the local communities to understand the research that we are doing. What's the significance to them? What are the tangible and intangible benefits that may accrue to them? So we say that first, we've got to do the research in the seaside. Then we bring it to the academe, where we are going to try to discover new compounds. And then eventually, we would like to bring our discoveries to development and get them to become drugs useful to our patients in the bedside. So let me walk you through what we call the drug discovery pipeline. It's a very involved process, but best of all, you must learn chemistry well. First, we have to collect the mollusks and the sponges with consent from the local communities. We bring this to the lab, we process the tissues, and then we isolate microorganisms from them and culture them in the lab. Step 2, we've got to test the microbial, mollusk, and sponge extracts for their bioactivity. So in this case, it's antimicrobial activity, anti-cancer activity, or neuroactivity. Third step, we've got to identify the isolates, the microbial isolates, or the sponge or mollusk organisms taxonomically. And the best way to do this now is through genetics. So we don't just do the classical taxonomy based on morphology. We've got to identify them through certain gene markers. Fourth, there's this challenging step where we have to do chemical extraction, chemical profiling, purification, and dereplication of compounds in these extracts. We've got to be sure that the compounds that we are pursuing are novel or new. They have not been discovered before. Fifth, the novel or new bioactive compounds have to be characterized fully. So drug discovery is very challenging, but it's very exciting as well. So what's this star tree here? This is what we call a phylogenetic tree. So in the background, you see the cartoon of the classical phylogenetic tree. But there at the center, you will see a tree that is based on the genetic markers of the organisms that have been identified. So it's telling you there are differences in the species, in the genera of these marine organisms. It's important for us to learn the genetics well. And more recently, we're also studying the genomics of whole microorganisms, because there are many markers that are important in the microorganisms, not just relating to taxonomy, but also relating to the compounds that they produce. Let's focus on a particular area of my research, and that is the discovery of neuroactive compounds from this group of mollusks called conoideans. So conoideans consist of cone snails. This is well studied by my mentors, Drs. Baldomero Oliveira and Dr. Lourdes Cruz, our national scientist, and I'm continuing the work. Aside from the cones, we have their older cousins, the turids. And the turids are more speciose. So there are about 12,000 species of turids, only 500 to 700 species of cones. Again, as represented here in a phylogenetic tree. So the title of this DOST-funded project is Anti-Pain and Anti-Neurodegeneration Drug Candidates, Discovery and Development. And my project collaborators are Dr. Cynthia Salama of UP Diliman NIMBB, and Dr. Aaron Joseph Villaraza of UP Diliman Institute of Chemistry. It's very challenging to study these turids because they're a very diverse group. They're mega diverse. There are a large number of unidentified species. They mostly inhabit deep tropical waters, and they are smaller in size compared to the better studied cones and augurs. And therefore, we require a greater number of snails per turid for peptide purification. So they're very beautiful. Actually, these turids, some of them are so tiny, you'd have to look at them under the microscope. And you'll know that each of them is different or unique in their shell morphology. But what's more interesting than that is that they have unique repertoire of peptides in their venom. And this venom is the source of our neuroactive peptides. Okay, so this is the overall strategy of the DOST funded project. It aims to discover, characterize, and develop anti-pain and anti-neurodegeneration compounds from Philippine conoideans and to test their efficacy in electrophysiology and dorsal root ganglion neuroactivity assays. I'll explain that a bit more later. So the three intersecting circles are to describe peptide discovery, so that's isolation of the peptides from the venom of the snails. Then because the amounts are so tiny, if we have a candidate neuroactive peptide, then we'd have to chemically synthesize the peptide, and that part is being done in Dr. Villaraza's lab. Another alternative is for the peptides to be biosynthesized through biotechnology in the lab of Dr. Saloma in NIMBB. Note that the asterisk in the intersection is common to all three circles. So for each of those activities, we got to do the purification, the characterization, and the bioassay. All right, so let's just look at one example of a turid. It's called Unidogemula bisaya from Cavite, and it's beautiful. You must say all of these snails are beautiful. So let me walk you through the methodology, and again, we've got to remind our scientists and researchers to learn the biochemistry and the neuroscience well. First, we have to do the crude venom extraction. Then we've got to do HPLC-based purification, bioassays. ICMBA there is intracranial mouse bioassay, and we've got the right permits to do this. And we've got self-based assays to help us identify promising neuroactive extracts that we would then subject to our second and third line assays. When we have a nice candidate peptide, we've got to produce more of it by synthesizing it. And because these peptides have a certain 3D fold, they've got to be folded properly and then purified and characterized. This is an example of an HPLC purification profile. And you've got a complex mixture of peptides there, and each of those peaks represents one or more peptides. The sharper the peak, the more pure that peptide is. So let me point out the arrow, which is UBIS3A. That's the one we pursue. So the first big graph is complex. We isolated that in the test tube, and then we run it again on the HPLC to produce that semi-pure peak on the right top part of the slide. Finally, we purified the peak as represented by that graph on the lower right part of the slide. We identified the peptide with this amino acid sequence. So UB3A has this amino acid sequence. Okay, so this peptide has a molecular weight of 1,798.52 Daltons, and it's got the unusual amino acid called a hydroxyproline. And what's more interesting is that when we injected the extract in 3-week-old mice, caused paralysis of the limbs within 20 seconds and death in 20 minutes. And when we injected the natural and synthetic pure peptide intracranially, there were tremors for 3 hours. So clearly the peptide is neuroactive. Okay, we have to compare the sequence of the peptide with other known amino acid sequences of the conoidean family of snails. It's just to give us an idea of their structure and their potential neuroactive targets. Okay, so there you have UB3A aligning with other peptides that had been isolated from conus snails. I'd like to show you some of the equipment that we acquired in the lab through the DOST project. So this is a cage that is used to house frogs, and it's a temperature, humidity controlled cage. And we need these frogs because we use their oocytes to express our neuronal receptors where we would test our peptides in. So this is what you call an electrophysiology setup. And what we do is to get the oocytes from the frogs, we put them under the microscope, we inject a DNA sequence for a particular neuronal receptor. It's then expressed on the surface of the frog oocyte, and then we're able to inject this oocyte with the peptide to see whether it affects the action potential that the receptor would be responsible for. Okay, that in a nutshell would describe the electrophysiology experiment. Okay, that's an example of a result of an electrophysiology experiment where the peptide would inhibit the action potential or the current through the ion channel receptor, which is the neuronal receptor. Okay, and the graph on the right side is called a dose response curve. It's telling you that as you increase the dose of the peptide, there is a change in the action potential or the current. It's telling you that this is a true biological response that is dose dependent. Okay? So that ends the first part of this talk on neuroactive peptides from conoideans like turids. I'll tell you now about the ICBG Philippine Mollusk Symbiote project that I have with our US collaborators led by Margot Haygood, and with her, Eric Schmitz and Baldomero Oliveira of the University of Utah. So many years ago, we hypothesized that these conus snails might harbor microorganisms in their tissues. So we started processing the tissues and growing microorganisms from them. And true enough, we got this whole slew of microorganisms found in various tissues of these 3 conus snails. And so this was the beginning of our USNIH-funded program called Philippine Mollusk Symbiote. And we were able to show that these microorganisms from tissues of mollusks, when cultured in the lab, would also produce neuroactive compounds. So this time we have here a streptomyces isolated from conus, producing small molecules called thiazolines, and they are neuroactive. How did we discover their neuroactivity? We used another kind of assay, but before I talk about that, I'd just like to show you another group of compounds isolated from another microorganism from the tissues of another snail. Okay, so in this case, we have here modified peptides. Not quite like the peptides from the venom of conus, because they are highly modified. Okay. So both groups of compounds are active in neuroassays. So let me tell you about the assay that we used to discover those two sets of compounds. It's called the dorsal root ganglion assay. So we get dorsal root ganglion neurons from mice, and we culture them in the lab, and then we monitor the entry of calcium into the neurons using a calcium imaging system. Okay, and this tells us whether the neurons are affected by the compounds that we have isolated. So this is an example of what you see in the microscope and the neuroactivity traces for each of the neurons that have been monitored through our calcium entry imaging system. We call this constellation pharmacology. Why? Because with this assay, we're able to capture activity in a wide range of neurons. So neurons from the dorsal root ganglion are heterogeneous. For simplicity, we classify them as small, medium, and large neurons, and we tag them in the plate where they're growing. And we're able to monitor what happens to each of these types of neurons when they are fed or exposed to the peptide, the compound, or even a crude extract. The traces below, from left to right, show us that a peptide could have an effect on the action potential through one type of neuron. Could increase, decrease the action potential, or have no effect on it. So take a look at the synthetic nobilimide B analogs that Dr. Villaraza synthesized. And it's showing an effect on the action potential through DRG neurons. Okay, so let's take a hold of this magnificent biodiversity that we have in our country. And I'm just focused on conioideans. We say we have a very rich biodiversity library in this mega diverse group of marine organisms. There are roughly 13,000 conioidean species, each producing about 200 peptides per venom. That's about 2.6 million peptides. And all peptides are already field tested. Why? What do the conioideans use these peptides for? They use them to hunt their prey, to avoid their predators, and also to compete with their competitors. plus, plus, plus. Why? Because now we're also discovering many microorganisms that live in the tissues of these conoideans, and these microorganisms are producing small molecules that are also neuroactive. But that's only the first part of the story. Why? Because these microorganisms found in the conoideans are prolific producers of anti-infective compounds. I'd like to acknowledge our funding agencies. That's the DOST, the US NIH, Fogarty International, National Science Foundation, and we'd like to recognize the contributions of our collaborators, both local and foreign. Most of all, we'd like to acknowledge all the graduate students and researchers who are embarking on this discovery of new drugs from the sea. Thank you.